Hi everyone. Today's lecture is on glycolysis part 2. The biochemical and physiological importance of anaerobic glycolysis. Glycogen to lactic acid. Type 2B muscle fibers, exclusively anaerobic, converts glycogen to lactate, generates all the ATP that is required to support the activity. Type 1 and type 2A muscle fibers use this process only when the demand for ATP exceeds that which can be generated from aerobic metabolism. Example, during hypoxia. Glycogen to lactic acid, fetus tissues. One condition in which all tissues or organs may use glycogen is the mammalian fetus during birth. This is especially the case if the placenta separates from the uterus prior to birth, which may occur if parturition is slow. In this situation, prior to birth and the first breath, the tissues of the fetus may generate ATP from conversion of glycogen to lactic acid. Survival depends on this process. To this end, the glycogen level in the tissues of the fetus increases just prior to birth. Lactate accumulation. Lactate released from cell undergoing anaerobic glycolysis is taken up by other tissues, primarily liver, heart, and skeletal muscle, and oxidized back to pyruvate. In the liver, the pyruvate is used to synthesize glucose via gluconeogenesis, which is returned to the blood. The cycling of lactate and glucose between peripheral tissues and liver is called the Cori cycle. The heart, with its huge mitochondrial content and oxidative capacity, is able to use lactate released from other tissues and Cori cycle. Glucose is metabolized via glycolysis in the muscle. A small amount of ATP is produced. Under anaerobic condition, pyruvate is converted to lactate. This lactate released from the muscles into the blood. Lactate is taken up by the liver and converted to glucose via gluconeogenesis. These chemical reactions require ATP. Glucose then released into the blood and taken up by the muscles. The whole process is known as Cori cycle. Lactate accumulation. In the muscle, glucose or glycogen converted to lactate via anaerobic glycolysis. This lactate transported out from the muscle using a lactate proton cotransporter. From the blood, the lactate is taken up by liver. In the liver, lactate is reconverted to glucose via gluconeogenesis. When lactate reaches heart oxidizes lactate aerobically to produce ATP and carbon dioxide. Usually, the Cori cycle occurs between the liver and the muscle. But there is a possibility for a Cori cycle to occur between liver and other tissues. Example, erythrocytes in their glycolytic pathway produces lactate as the end product. This lactate can be circulated back to liver and then again converted to glucose. Likewise, in the bone marrow, glucose anaerobically converted to lactate. Again, this lactate taken by the liver and converted to glucose. In the lymph node, the lactate derived from glucose taken by the liver again converted to glucose. These Cori cycles are referred as extended Cori cycle. Lactic acidosis. The concentration of lactate in the blood is a function of both tissue lactate production and lactate consumption. Compared with daily lactate production, the amount of lactate that circulates in the blood is very small. Hence, even a relatively small change in production or consumption can have a sizable effect on the plasma concentration of lactate. Hyperlactemia is a condition of an abnormally high concentration of lactate in the blood, which is typically defined as greater than 2 millimole lactate. Normal reference range value 0.5 to 1.5 millimolar. Major tissue sites of lactate production in the resting man. An average 70 kilogram man consumes about 300 gram of carbohydrate per day. Daily lactate production gram per day. Total lactate production is 115. Red blood cells produces 29 gram per day. Skin produces 20 gram per day. Brain 17. Skeletal muscle 16. Renal medulla produces 15 gram per day. Intestinal mucosa 8. Other tissues 10 gram per day. Lactic acidosis. Lactic acidosis develops when lactate production exceeds consumption such that the pH in the blood drops and the concentration of both bicarbonate and carbon dioxide become abnormally low due to buffering and increased ventilation. Lactic acidosis is typically seen in when the blood lactate concentration is 4 millimolar or greater. The term anion gap refers to the difference between measured serum or plasma concentration of cations, sodium and potassium. Potassium is often omitted, and the anions chloride and bicarbonate. The anion gap, therefore, an indicator of the concentration of unmeasured ions, lactate, acetoacetate, beta-hydroxybutyrate. A patient who has lactic acidosis may have an increased anion gap. Effect of hypophosphatemia in glycolysis. The term hypophosphatemia refers to an abnormally low concentration of phosphate in the blood. Hypophosphatemia impairs flux in glycolysis by reducing the activity of glyceraldehyde 3-phosphate dehydrogenase, which converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. In tissues with mitochondria, hypophosphatemia also impairs ATP production by oxidative phosphorylation. Tissue oxygenation suffers because red blood cells contain less 2,3-bisphosphoglycerate to facilitate the dissociation of the oxygen from hemoglobin. Prevention of dental caries with fluoride. Dental caries is caused by Streptococcus mutans. This bacterium glues itself to the tooth with the help of a polysaccharide that makes from sucrose and generates its energy by turning glucose and other sugars into lactic acid. The lactic acid erodes the acid-sensitive calcium phosphate in the tooth enamel, causing cavities. Both bacterial and the human varieties of the glycolytic enzyme enolase are inhibited by fluoride ions, a common ingredient of toothpaste. Tumor hypoxia. When a small localized tumor outgrows its vascular supply, tumor hypoxia arises in regions with impaired oxygen delivery. The alteration of gene expression in tumors because of hypoxia. The hypoxia that some tumors experience with rapid growth activates a transcription factor, hypoxia-inducible transcription factor, HIF1. HIF1 increases the expression of most glycolytic enzyme and the glucose transporters GLUT1 and GLUT3. These adaptations by cancer cells enable a tumor to survive until blood vessels can grow. HIF1 also increases the expression of signal molecules such as vascular endothelial growth factor that facilitate the growth of blood vessels that will provide nutrients to the cells. Aerobic glycolysis is a property of rapidly growing cells. Tumors can be visualized with FDG, a non-metabolizable glucose analog, and positron emission tomography. The figure A indicates the pre-therapy, a non-metabolizable glucose analog, FDG, infused into a patient and detected by a combination of positron emission and computer-aided tomography reveals the presence of a malignant tumor. After 4 weeks of treatment with a tyrosine kinase inhibitor, the tumor shows no uptake of FDG, indicates decreased metabolism. Excess FDG, which is excreted in the urine, also visualizes the kidney and bladder. Entry of other sugars into glycolytic pathway. Galactose converted to UDP galactose and then UDP glucose. UDP glucose converted to glucose 1-phosphate, and glucose 1-phosphate enters the glycolytic pathway at the level of glucose 6-phosphate. Mannose converted to mannose 6-phosphate by hexokinase and then enters the glycolytic pathway at the level of fructose 6-phosphate. Lactose converted to glucose and enter the glycolytic pathway at the level of glucose 6-phosphate. Sucrose converted to glucose and fructose and enter the glycolytic pathway at the levels of glucose 6-phosphate and fructose 6-phosphate. Fructose via fructokinase pathway converted to fructose 1-phosphate. Fructose 1-phosphate cleaved into glyceraldehyde and dihydroxyacetone phosphate, and these two can enter the glycolytic pathway at the level of glyceraldehyde 3-phosphate. Polyol pathway and its role in disease. The polyol pathway consists of 2 steps. Glucose is converted to sorbitol and sorbitol is converted to fructose, and it is present in a wide variety of cells. Sorbitol is a sugar alcohol. Aldol reductase is the rate-limiting enzyme of the polyol pathway. It has very low affinity for glucose. Thus, the flux in the pathway is markedly increased during hyperglycemia. Polyol pathway and its role in disease. In diabetic patients who have chronic hyperglycemia, activity in the polyol pathway is thought to contribute to lens cataracts, peripheral neuropathy, retinopathy, and nephropathy. Increased aldose reductase activity lowers the concentration of NADPH and thus lowers the concentration of reduced glutathione. It permits the presence of increased amounts of reactive oxygen species, which then inflict damage to various cell components. Sorbitol may accumulate in tissues that have low sorbitol dehydrogenase activity. This may damage cells through osmotic effects and contribute to neuropathy, retinopathy, and the formation of cataracts. Complications of diabetes, in part due to increased flux through the polyol pathway, are shown in the figure. These are proliferated diabetic retinopathy, peripheral neuropathy, and diabetic nephropathy. And you can see in this figure the difference between a healthy eye and an eye with a cataract. Polyol pathway in seminal vesicles. In the evaluation of infertility, fructose is often measured in the ejaculate. About 90% of the fructose in the ejaculate is derived from the seminal vesicles, which synthesize fructose via the polyol pathway. The concentration of fructose in semen, normally more than about 7 millimolar when measured 1 hour after ejaculation, thus serve as an indicator of the function of the seminal vesicles. In the absence of fructose, sperm have a much shortened lifespan. Spermatozoa use fructose as a major fuel source. Spermatozoa use fructose as a major fuel source while in the seminal fluid and then switch to glucose once in the female reproductive tract. Utilization of fructose is thought to prevent acrosomal breakdown of the plasma membrane while the spermatozoa are still in the seminal fluid. Sites of problem with fructose metabolism. Fructose malabsorption. Deficiency of fructokinase leads to essential fructosuria. And deficiency of aldolase B enzyme leads to hereditary fructose intolerance. Hereditary fructose intolerance. Aldolase B deficiency, it is an autosomal recessive inheritance. Accumulation of fructose 1-phosphate. Cytosolic ATP is utilized for the formation of fructose 1-phosphate. There will be a deficit in cytoplasmic phosphate. Insufficient ADP is phosphorylated to ATP. ADP and AMP are degraded to urate, which is released into blood. Marked depletion of ATP in the kidneys and liver causes failure of these organs. In the kidneys, the proximal tubules fail to take up electrolytes from the glomerular filtrate, a condition called renal Fanconi syndrome. Fructose-induced hypoglycemia due to excessive stimulation of glucokinase, inhibition of glycogenolysis and gluconeogenesis. Concerns about high fructose consumption in the general population. Fructose consumption of 100 gram per day or more is associated with hypertriglyceridemia and often with hypercholesterolemia. Fructose metabolism can feed trioses into glycolysis at a greater rate than can occur with glucose. Then pyruvate and citrate are also produced at high rate. Fatty acyl-CoA are synthesized at high rate, and fatty acyl-CoA are readily esterified with glycerol 3-phosphate, thereby giving rise to triglycerides. High fructose intake is also associated with gout. High intake of fructose from sucrose, high fructose corn syrup, fruit juices or fruit can lead to an accumulation of AMP in the liver and kidneys. AMP is then degraded into urate, which can lead to hyperuricemia, a precondition of gout. Galactose metabolism. Galactose first converted to galactose 1-phosphate by galactokinase. Galactose 1-phosphate combines with UDP glucose to form UDP galactose and glucose 1-phosphate. Glucose 1-phosphate is converted to glucose 6-phosphate by phosphoglucomutase, and subsequently glucose 6-phosphate enter the glycolytic pathway. The diseases associated with galactose metabolism. Classic galactosemia. Galactose 1-phosphate uridyl transferase deficiency. It is an autosomal recessive disorder causing galactosemia and galactosuria. Accumulation of galactose 1-phosphate and galactitol in the liver, brain and eyes causing liver damage, brain damage and cataracts. Prenatal diagnosis possible by chorionic villus sampling. Newborn screening also available. Treatment, removal of galactose from the diet. Galactokinase deficiency is a rare autosomal recessive disorder causing elevation of galactose in the blood, causing galactitol accumulation if galactose present in diet, elevated galactitol causing cataracts, treatment, dietary restriction. Lactose synthesis in the lactating mammary gland. Glucose is converted to glucose 6-phosphate, and then glucose 6-phosphate is converted to glucose 1-phosphate. Glucose 1-phosphate combine with UTP and form the UDP glucose. UDP glucose converted to UDP galactose by UDP galactose 4 epimerase. UDP galactose combined with glucose in the presence of lactase synthase synthesizes lactose in the mammary gland. Thank you.